Welcome to the Herts and Essex virtual taster session for A-Level Biology.
I'm Miss Johnson and I'm Subject Leader for Biology. During this taster session, I'm going to talk through the course, we're going to hear from our current students, and I'm going to give you an insight into what a lesson might be like. But first, let me introduce the biology team.
We have Mrs. Dalton, who's also Head of Science, Miss Jones, who's also Head of Year 9, Mrs. Wright, Dr. Spillett, and Mrs. Ruthven. The exam board that we follow at A-Level is AQA. We've picked this exam board because they provide us with a range of resources that we can use in lessons, like exam questions, and it's really important to embed that exam technique early on. The course also has a practical endorsement element to it. So there isn't any practical coursework, but there is a big emphasis on building those practical skills. And university courses will look for courses that have practical endorsements associated with them. So it's a very desirable characteristic to have. And there are 8 topics that we cover over the 2 years. 4 are done in the first year and 4 in the second year. And you can see a list of those topics on the PowerPoint. Those topics have subtopics within them. So if we take number 2, cells, we will start off looking at the different structures within cells and then move on to transport across the cell and finish off with immunology. Some of these topics will build on your GCSE knowledge and some of them will be completely new content. This is an overview of the assessments that you will do for the course. So there are 3 papers that you will take at the end of 2 years. The first paper is on your Year 1 content, the second paper is on your Year 2 content, and paper 3 is a synoptic paper which draws links between several topics across the 2 years. There is a range of skills that are tested on, so you'll get questions about the practicals that you've done, there are comprehension-based questions, and you can see in paper 3 that there's also a synoptic paper, a synoptic essay at the very end. So why study A-Level Biology? Well, first of all, it's fascinating. At A-Level, we really do get into the nitty-gritty of how living organisms work. The course also develops a range of skills, including problem-solving skills. We develop your written and verbal communication of the content. We build your data analysis skills by looking at graphs and data tables, as well as build those practical skills, which I mentioned earlier. These skills can be applied to a range of career paths. So you can go into healthcare professional careers, but you can also go into lots of other careers as well. So it really does open the doors for you. And we're really proud at Herts and Essex of our outstanding results that we get. So for A-Level Biology, we get 7% A star to B. It is a really popular course. And so we ask for a minimum of a grade 7 in biology GCSE or a 7/7 if you're taking combined science. We also ask for a minimum grade 6 in maths because 10% of the marks in the exam are maths-based questions. If you do get a grade 6 in maths at GCSE, we will also support you with a core maths course as well. If you get a grade 7, then you do not need to take that core maths course, but we will support you with those math skills within lessons by making sure that we do lots of practice, as well as giving you a massive biologist booklet at the very start of the course. So we're going to hear now from one of our current students to find out why did they decide to do A-Level Biology and what they have enjoyed learning about so far.
Hi, I'm Grace and I do biology, chemistry and history at A-Level. I decided that I want to do biology because it's the most interesting, as I love learning about how the body is able to adapt and function to survive. My experience of A-Level biology at Herts and Essex has been a very positive one, and I would say it's my most favourite subject. My favourite topics that we have done so far are DNA, because I love learning about the coding of DNA and the replication, and also the immune system, so how the body fights against diseases. This is because in the future I'm looking to go into a course in genetics or the development of drugs, and this has given me a really helpful insight into these. I also love the fact that you can do practicals in biology, because this is a really fun and useful way to visualise what you have been learning, and you get to use really cool equipment like the centrifuge as well that you learn about. My favourite practical has been the heart dissection, and I wasn't too sure about it at the start. I was a bit squeamish, but once you got involved and looked at all the different sections of the heart, it was actually really fun and definitely my most favourite. I would highly recommend doing biology for A level because it is really fun and interesting, and all the teachers are very helpful.
We carry out a lot of practical work at A level, but as part of your practical endorsement, you will complete a minimum of 12 compulsory practicals over the 2 years. So there are 6 that you do in your first year and 6 that you do in your second year. We do way more practicals than just 12 because we want you to get experience of developing those practical skills and those investigation skills. The main focus of these practicals is for you to get a range of techniques and get used to using a lot of different pieces of equipment. In your exams, you will be tested on those techniques and the equipment that you have used, and that will equate to about 15% of the marks in the exam. We will give you a lab book where you will record and put evidence down for the techniques and equipment that you have used and to show your skills developing over those 2 years. And we're going to hear now from one of our students to find out how they have found those practicals.
Hello everybody, my name's Sam. I'm currently studying biology, chemistry, maths and PE. Basically throughout the year we do various different required practicals. So far we've done dissections, we did a fish gill and a heart. We've also done experiments about permeability of beetroot and osmosis in potatoes, which is fun. Personally, my favourite ones are the dissections. It's interesting to see, the theory side come into the practical side and like cutting open the heart and seeing what you've learned about. You're also assessed on various different criteria during the practicals, such as safety and techniques. So yeah, overall it's pretty fun. It's a good break from the theory side. So yeah, that's practicals for you. Hope everyone is doing well. And yeah, back to you in the studio, Miss Johnson.
We're now going to look at an example investigation. So we're going to look into the effect of varying the concentration of the substrate, hydrogen peroxide, on the rate of a reaction catalysed by catalase. So this is all to do with enzyme activity. So before we look into the investigation, let's go over some of the theory. So here is a summary paragraph of how different factors could affect enzyme activity. Have a go at filling in those numbers with the key terms at the bottom. And we'll go through the answers in a bit. This is a good chance to pause the video to see if you can work it out. Here are the answers in green. Hopefully these keywords are familiar to you, but as a recap, enzymes speed up reactions. They don't get used up, they just speed up a reaction. The reactants in the reaction are called a substrate, and enzymes work specifically on one particular type of substrate. Different factors can affect enzyme activity, and those factors are enzyme concentration, substrate concentration, temperature, and pH. When those conditions are optimum, it means that enzyme activity will be really high, it'll be really fast. But if those conditions are not at their optimum, it could decrease enzyme activity, and it could also stop the enzyme working completely, and we call this denature. So that's a summary of the theory. Now let's try and apply that to our investigation. So our investigation was looking at changing the substrate concentration. Our substrate is hydrogen peroxide, and there are 5 different concentrations that we're going to test. 0, 5, 10, 15, and 20 vol. Vol is our unit for concentration. So we've got 5 different concentrations, and we're going to see how that affects our enzyme activity. Our enzyme is found in potato, and it's called catalase. So looking at our experiment setup, we've got a burette here that's turned upside down and it's full of water. That burette is connected to a test tube via a delivery tube. Inside that test tube is our substrate and our enzyme. What will happen is that hydrogen peroxide will break down, and when it breaks down, it will release oxygen as a product. And you can see the bubbles of oxygen going into the burette. What they will do is they will then displace the water. So the volume of water that is displaced is equal to the volume of oxygen that is given off. And that will tell us what the enzyme activity is like. So if the oxygen volume is given off as high, then it means that the enzyme action was very high. When we're planning an investigation, we need to consider certain variables, and you would have done this at GCSE. So the first thing we consider is our independent variable. What is the one thing that we are changing in our investigation? So we're doing 5 investigations. What is the thing that is different each time? That is our independent variable. The second thing that we consider is our dependent variable. What are we measuring? And this is the outcome of our experiment. It is dependent on our independent variable. So what is it that we are measuring? And then lastly, when we're planning an investigation, we need to consider our control variables. And there are always a lot of these because they need to be kept the same to ensure that we collect data that is reliable. So why don't you pause the video, see if you can think about the answers to these questions. What are our variables for our investigation? Here are the answers in green. So our substrate concentration is our independent variable. We've got 5 different substrate concentrations, 5 different concentrations of hydrogen peroxide. We are going to measure the volume of water displaced, or we could have also said the volume of oxygen given off. They mean the same thing. That is our dependent variable. And then lastly, we've got all our control variables. There is a long list here. These need to be kept the same to minimize errors throughout our experiment. So we're going to be doing different hydrogen peroxide concentrations and measuring the volume given off. We need to be measuring for the same time. So we're going to do that for 2 minutes. Our concentration of our enzymes should be kept the same. Our surface area of our potato should be kept the same. The number of discs that we put in should be the same. So we're going to do 5 discs for each of the 5 experiments. The volume of solution is going to be 10 mils. So that's our hydrogen peroxide volume and our discs together. Our conditions should be kept the same. For example, temperature and pH, because if they're not kept the same, then they could also affect enzyme activity. And it's also important that we don't stir because that reaction mixture can get mixed up a bit more and that could affect the volume of oxygen being given off. So that's our planning. Now let's have a look at some data. So this is some example data of what we could have got from the experiment. We've got our burette reading. So it shows what the water level was like at the start of that experiment and what it was like at the end. I want to see if you can work out what was the gas produced. So if we know what our water level was like at the start and the end, then we can know what our gas is produced. I'd also like you to have a go at working out our reaction rate for our experiment. So this involves a bit of maths. So we did our experiment for 2 minutes. So each concentration was done over 2 minutes. So use your gas produced divided by 2 minutes to get the volume of oxygen per minute. Once you've got that, then I want you to see if you can work out what hydrogen peroxide concentration was used for each beaker. So let's say the beakers weren't labelled and we need to try and find out what they were. Well, try and use the data to work out what that hydrogen peroxide concentration is. And then see, once you've put that in, what is the trend in our data? So as you increase hydrogen peroxide concentration, how does that then affect the gas that's being produced? And then see if you can then write an explanation using scientific knowledge. So using the information from the paragraph earlier, what is the link between hydrogen peroxide and gas given off? Why does that happen? See if you can add an explanation. So again, pause the video, have a go at answering those questions, and then we'll go through it. So here are the answers. Did you manage to do your calculations correctly? Have you managed to work out what hydrogen peroxide goes with which unknown concentration in the beakers? This is an example of what we would typically do in a lesson. It's really important to build those planning skills. It's important to be able to do our math skills as well as data analysis and look at some data. So this is very typical of a lesson. We also do a lot of exam practice in lessons, as I said, to build that exam technique. So here's an example of an exam question to do with enzyme activity. Again, you can pause the video, stop it, see if you can have a go at writing your answers to this question. And I will show you the mark scheme in a bit. Here is the mark scheme. Each question was worth 2 marks. You can see the points for each marking point on the mark scheme, and they're separated by a semicolon. To finish off, we're going to get some advice from one of our current students, Roz, to see what her top tips are for studying biology at Herts and Essex. So here are her top tips. She says, keep your GCSE revision guide. I think that's a really good top tip. Print off the specification for the course. So we do AQA and we will give you the specification for you to print off at the start of the year, and it's a really good checklist. Roz also says, keep your folder as organised as possible. Organisation is key. Ask questions. I couldn't agree more with that one. We love hearing questions from students. It's really important to deepen your understanding and to ask us lots of questions. So please do that. Number 5, she says, cement your learning. So we don't do much note taking in lessons. And so doing that after lessons helps embed some of that knowledge that you've learnt. And lastly, practice exam questions. Again, this is key. We do this from the get-go. It's really important to build that exam technique earlier on. So we use exam questions in lessons, but also use it as a revision tool as well. I hope you have enjoyed your virtual taster session for biology. Thank you for listening and looking forward to meeting you all.